what lights their fire, why they're excited about what they're doing, and where they see it going. And the first speaker is Kim Lewis. He's a distinguished professor, University Distinguished Professor in the Department of Biology. He's also the director of the Antimicrobial Discovery Center. Kim has a PhD and bachelor's degree from Moscow University in the USSR, and his area is microbiology. So at that point, I'd like to have it with Kim.
Well, thank you, Maury, for the introduction, and thank you all for coming here. And I will tell you about what lights my fire, and I hope that the results of our work will speak for themselves. But by way of a short introduction, I will say that I've always been interested in biology as a whole. And that, of course, leads you to microbiology, because microbes interact with each and every creature on the planet. And we now live, I think, in the time of microbes, because we know from the revolution that is going on in the microbiome field that microbes affect most, if not all, functions of our body and mind. And we also are in a very serious standoff with our pathogens. So it is these two areas that my lab works in, and I will tell you about that. If there is a common theme in the things that we do, it is not any particular subject, but it is something that we all enjoy doing, and that is attacking old unsolved problems and paradoxes. Why? Because it is fun. So here's a paradox, a good one. It goes like this. A paradox of chronic infections. Antibiotics have limited efficacy against antibiotic-susceptible cells. And if that makes no sense to you, that means you have been paying attention. So what we discovered studying chronic infections is that actually antibiotics do pretty well in killing the majority of cells. And then there's a small minority that survives. These are persisters, and of course it is nice to know why they survive and what are they. These are not mutants, these are variants of regular cells. So we decided we'll isolate persisters and find out something about them. But it is kind of difficult to isolate something if you know absolutely nothing about it. So then we decided we'll base isolation on a guess. And the guess is that persisters are dormant cells. And if they are dormant, then we can use this trick. We can use a strain of E. coli that carries a gene coding for a degradable green fluorescent protein. So if your population is growing, old cells are making it rapidly and they are green. If one of them decided to go into dormancy, it goes dim, because it doesn't make new one and the old one degrades. So then we expose this culture to a cell sorter and see that it indeed splits into two. Here is your bright majority, regular cells, and a dim minority. So now the sorter will sort them into a test tube. You expose them to antibiotic, and you see that they are considerably more tolerant to killing than the regular cells, which we now renamed, and these are now quitters. So having them in a test tube allows us to do transcriptome analysis, to ask the question, what are the genes that are specifically turned on in these cells? And we are looking for genes that code for proteins that could shut down important functions in the cell, causing dormancy. So here are these candidates. These are all toxin, antitoxin modules, which nobody knew what they are doing in the cell. Now we do know that. Toxins, antitoxins are persister genes. They code for dormancy programs. So by different mechanisms, they shut down either protein synthesis or deplete cell of ATP. That causes dormancy. And there are more than 10 of these pathways. So while it was very satisfying to find this and interesting science-wise, that is not terribly useful to discover drugs that are going to treat chronic infections. Because, here are dormant cells. They have these parallel pathways. You cannot knock all of them out. Antibiotics only act against active targets that require energy, and these guys are energy low. So this looks like a perfect defense, and we are in a perfect dead end. So when you find yourself in a dead end, I find that it is very useful to ask a question. What is it that you want in a perfect world? So in a perfect world, we're looking for a compound that is going to hit an important target in the cell, corrupt it, so that it produces some toxic product, and that is not going to require energy. Right? So that is our fantasy perfect compound. Well, once you formulate that, actually that helps you zero in on this old abandoned antibiotic, acyl depsipeptide. It does a very interesting thing. It activates this protease in the cell. The protease would normally degrade misfolded proteins. It requires ATP, but in the presence of this compound, the pore of the protease opens and it can degrade peptides without an ATP required. So we thought that is a very interesting candidate for what we're looking for. And so we did a simple experiment. Add a regular antibiotic to cells, and you see this biphasic killing with surviving persisters. If you add ADEP, persisters collapse, there's complete sterilization. We're very excited because it was the first time that we could sterilize an infection without using bleach. So then we tested it in an animal model. This is a massive infection by MRSA Staph aureus. Regular antibiotics do nothing in this model. And if you add ADEP in combination with another antibiotic to prevent the rise of resistant cells, you get complete eradication. So now we have a sterile mouse, a new creature, if you will. We're developing this in a therapeutic, and I think this will help treat untreatable chronic infections. They're untreatable today. So another thing I will tell you about, another old unsolved problem in microbiology, is uncultured bacteria. The majority of bacteria in the environment are uncultured. And here's a simple experiment. You take a sample from marine sediment, for example. You put a droplet under the microscope and you count cells. And then you put another one on a petri dish and you count colonies. And the difference between these two counts is known as the Great Plate Count Anomaly, described by Winterberg in 1898. So a number of years after Winterberg, my colleague Slava Epstein here from Northeastern and I decided that we'll come up with a method to grow uncultured bacteria. Our idea was very simple. We will grow them in their natural environment because, of course, that's where they do grow. And so then the question is how to come up with a gadget that will allow you to do that. So here's the gadget. We take a sample from marine sediment, dilute mix with agar, put it between these two semi-permeable membranes which go on this O-ring. We buy it at Home Depot. And then this whole contraption, which we call diffusion chamber, goes back into the environment. Now everything diffuses through the chamber and the bacteria are tricked into perceiving it as their own habitat. And all kinds of creatures grow, including this very important one. I have no idea what it is. And from a variety of these organisms, we can then isolate new antibiotics. So that's the applied side of this project, and we're collaborating with Novabiotic Pharmaceuticals, a biotech startup, to do this. So here's an example of such a novel compound. It's an antimicrobial. We isolated this from a previously uncultured bacteria, gave it this name, Eleftheria terra. Eleftheria means freedom in Greek. You will see why we gave it this name in a moment. So we tested it for resistance. It kills a lot of pathogens, and there's no resistance. So then we discovered a new detergent, because that's what detergents do. They dissolve membranes, and there's no resistance to detergents. Very boring, because we also have membranes, of course. So, but then we took this compound and exposed it to mammalian cells, and there was no activity. So it's not a detergent, and there's no resistance. So that was very intriguing. We decided to figure out the mechanism of action of this compound, and what we found out is that it hits targets which are not proteins, so they do not mutate. it hits this compound, which is a polymer precursor of peptidoglycan in the cell wall, and another polymer, which is a precursor of another cell wall polymer, wall-teichoic acid. So it hits two independent targets. Neither of them is a protein. Neither of them can mutate, and there's no resistance. So I think this tells us that the dogma that bacteria will always develop resistance to antibiotics is simply incorrect. All right. So, of course, apart from finding antibiotics, we were very intrigued by why uncultured bacteria don't grow. We had a guess that perhaps they do not grow because they require growth factors from their neighbors. And here's a test of this hypothesis. So you take marine sediment, you dump a lot of it on a plate, and a whole zoo of colonies grow here. And then we say, okay, maybe this little colony grew because it was next to this one, which was producing a growth factor for it. So then we test that by plating these two on the same petri dish. And here's your cultivable organism, and the other one is spread evenly, and it grows only around it, right? So clearly this creature is making something, a growth factor. So then we grow a lot of this, fractionate the growth media, add the fractions one by one, and ask the question, in which fraction do we have this active principle? We do that in collaboration with John Clardy from Harvard Medical School, and this gives us our first growth factor. This turns out to be a siderophore, a chelator of iron. So bacteria release siderophores from the cell. They go out there, grab iron, and bring it back to the cell. Uncultured bacteria lost their ability to make siderophores, and with that, lost their liberty. Why they did that, I have no idea. This is an interesting mystery. That's sort of the next thing to figure out. So armed with this experience, we decided to go into human microbiome, into our guts, and look for growth factors for uncultured bacteria for the human microbiome. And I'll tell you what we found. So first of all, we found the same principle. There are lots of uncultured bacteria that will be helped by other bacteria. So here are these pairs. I will tell you about this one. So here's a Doria longicatina, a cultivable organism that helps Flavonifractor to grow. Flavonifractor has been for a long time on the NIH most wanted list. Most wanted list of not sort of criminals, but bacteria that we have been unable to culture in the lab, and we know about them only by their genomic signature. So now we can grow it, and we isolated together with John Clardy the growth factor, and here it is. This is a very interesting thing. This is GABA, gamma-aminobutyric acid. You probably know that it is an important neurotransmitter. Okay? So we tested the ability of this creature to eat a lot of nutrients. Here is the list. The only thing that it can survive on is GABA. So then we sequence this genome. All the entry points into general metabolism are absent. The only entry point is from GABA. So this creature evolved to eat only one thing, our very important neurotransmitter. So what does that mean? Well, I think one of the possibilities is this is a depression bug. If we don't have a lot of GABA, we go into depression and anxiety. And that's the only thing that it eats. So that tells us that here is an opportunity to do something about it. And we right now, in collaboration with Lisa Barrett from Northeastern, are using Northeastern students as a very large group of sophisticated guinea pigs to study the correlation between their microbiome and the mental state, such as anxiety and depression that you guys, some of you get during exam periods. We pay $20, by the way, for that service, so keep that in mind. So I think that will tell us something very important. That will tell us about the correlation between the composition of our symbionts and pathobionts and our mental state. And this is, I think, a good example of how you sail out trying to address an esoteric problem of growth of marine sediment bacteria and end up contemplating the nature of depression and anxiety. And that is what science sometimes presents to you as an unexpected gift. Thank you.
Thank you very much. We're going to go through 4 talks in an hour, so we don't have a lot of time, but I'd be willing to take a question. Someone has one? I guess I'm going to ask one then, Kim. If you manage to wake up persisters and kill them so that problem goes away, how are bacteria going to evolve with another defense mechanism? What's your prediction?
That's a good one. Well, what they will do most likely, they will develop resistance to that particular compound with which I'm hitting them, and then I'll have to come up with another one, right? And we'll see who runs faster.